The chairman of Immunovia has claimed that early diagnosis in cancer may save more lives than therapy. Our next speaker will talk about their innovation and about their collaboration with academia. And I may add that this Swedish company, Immunovia, last year won an European Innovation Prize. So please welcome Laura Chirica.
Ladies and gentlemen, thank you very much. I'm looking for Morten to thank him for inviting us here, and thank you for the introduction. Well, as you can probably see, understand, I'm not Swedish, but I'm really very proud to be representing Immunovia today. I'll just leave the slide here, and I'll show it to you in a couple of minutes. Our talk today is going to present, as Nadia said, a breakthrough innovation, which is an antibody-based technology that was developed at Lund University by Professor Carl Borreback and his team, and is now in the market introduction commercialization by Immunovia. A large part of the talk is going to be dedicated to the first test, which is based on the technology. And this test is going to address one of the most important unmet clinical needs in cancer for one of what many people call in these circles the deadliest cancers of them all by mortality, which is pancreatic cancer. I would like to start by asking you one thing. How many of you, just raise your hand, this way you will also wake up. How many of you have any connection whatsoever with pancreatic cancer? Know a lot about it or have relatives or anything like this? I just want to make a parallel. Oh, fantastic. Okay. That's very, very good because there are several studies that are conducted in the UK, in fact, where there are about 60 to 70% of all the people, they know nothing or almost nothing about pancreatic cancer. You are much, much better. Before I am going to move on to the presentation, I just want to dedicate this talk to Professor Rosling for many, many reasons, for inspiring us, for his enthusiasm, for his passion, for presenting data and numbers like nobody ever done before, and for providing us with hope, which is what we would like to do with our platform and our test. So you see here, I just picked this particular part of his talk, where he says, this is Sweden, and next to it is actually Norway. So he's just pointing to us. How am I going to present today? I'll just give you, we have a short time, so I'll just take you through all of this. And just hang on, and I'll come to the results, which are the most exciting part of everything. I'll just show you two numbers that I want you to remember about pancreatic cancer. I'll take you through the company very quick, and I will take a longer time to present the technology. And I will also show you the latest results, because they are the most interesting of them all. I'll show you also a number of the clinical uses that have been developed in collaboration with several key opinion leaders around the world. And I will also show you where we are at this point in time with what we are doing at Immunovia. And, of course, we have to end up with a look into short-term, mid-term, I don't dare to look long-term future. I want you to remember about pancreatic cancer, the number 5. And why is 5? Because it is the survival rate for 5 years is 5%. So 95% never make it to the 5-year. And the other, I will show you in one slide the other number 5 that I want you to remember. Before that, just why I say that many people consider it the most deadly cancer. Here you see the data that are coming directly from the cancer registry in Sweden, where we can see the diagnosed and the diseased samples, the patients here, that were coming from a screening program from 2007 to 2015. You see the relation, right? I'm adding on top of it the breast cancer, which we also have screening. I don't know where Bjorn Hoffman is, who was asking us if we look for the healthcare or we look for the trouble. So we definitely look for providing benefits. And this is pancreatic cancer, where the number of people who get diagnosed is equal with the number of people who die. What you see here, the gap that you see between the two, is because they die before they are diagnosed. So they end up in another registry. But the number of people that are diagnosed is equal with the one that die. Now, we heard from Nadia, early diagnosis is the hope. And because of the nature of the disease, because of the development, because of many, many reasons, that is absolutely true at this point in time. We talk about early all the time. We say early. So what is early in pancreatic cancer? And there are a number of definitions around, and we chose the international guideline that says that the early detection is considered to be when the cancer is resectable by surgical procedure. And that holds true for stage 1 and stage 2, where we also see the stage 2 where it goes into stage 3 where it's not possible anymore, but it's stage 1 and 2 that we consider early stages. I included one very, very important study that is used very much today and referred extremely much from Japan, where pancreatic cancer has the highest incidence in the whole world, double of what we have in our countries, where they looked at 10 years. This is real-time study, 10 years, and they found that stage 1, 1A, 1B, 2A, 2B, they are the ones where the survival rate, if found early, goes up to 50 to 60%. So we have a tenfold if we detect it early. Today, the diagnosis is much too late. You know, Hans Rosling had 1 year, which is anyhow much longer than the number 5 that I have second time, which is not even 5, it's a 4.6 month of survival for anybody who is diagnosed with pancreatic cancer in Europe. Okay, this is the median one. Now, we switch gears and we look very quick at what is Immunovia. And, you know, we are coming from a very, very rich, very important heritage from Lund University, from Create Health, from the Department of Immune Technology, where antibody development has been one of the focuses for over the last 20, 25 years. About 75 to 100 researchers work with this platform still. Now, the possibilities that we have in Scandinavia where the patents are placed in companies that was discussed in the morning in different presentations, how important it is to have the industry taking over the innovation and bringing it to the market. We are taking on the tests and the discoveries and taking them through development, validation, and bringing them to the market. First test, IMREN-PanCan-d. But because of the nature of this platform, we are also able to develop diagnosis, prognosis, differential diagnosis, therapy monitoring also is possible, and there are research studies being done where we can see autoimmune diseases as well as cancer and different types of cancer. So we have a pipeline of a number of different tests. Well, where we are right now, we are in the market introduction of IMREN-PanCan-d, a lot of efforts being put into the verification, validation for the introduction to different markets around the world. We work extremely hard on getting our laboratory in Lund to be up and running, to be able to receive the samples, have a certification, ISO certification, global accreditation, and we also partner with different reference laboratories around the world. We have one technology transfer to Oregon Health and Science University in the US. I'm going to show you in some slides the results from that collaboration. So, I will take you through now the platform and just the most important parts of it that I would like you to remember when you hear about biomarker signatures, as you heard a lot during today. The hypothesis from the very beginning has been that blood contains enough information early enough to be able to, create some kind of a snapshot of the immune system response. This snapshot is, in fact, also called a disease fingerprint because it is specific to that particular disease. And these are the ones that we call biomarker signatures. In this case, we were looking to target the high-risk individuals. This was also one part that I enjoyed very much from the ethics talk this morning, where it was a lot of focus on having an approach on high-risk individuals and looking for that in providing the benefit to healthcare. And this is what we have been also focusing on, looking for asymptomatic individuals. Now, how do you build the biomarker signatures? And there are many different ways today. And as you heard in the morning, after the genome has been available, then lots and lots of different kind of technologies have been developed. And there you will see biomarker signatures where, in our case, is everything related to cancer, cytokines, complement proteins, and so on. Now, our platform, here you see 25 years in just one slide. And we have one part which is the production and the other which is the analysis. On the production side, it's very important to remember our antibody library, which is unique and proprietary. It has been engineered and it's been taken just one part of the antibody, which is the single chain fragment variable. It's in this way avoiding the problems with stability, with epitope activity, with binding to the surface, with reproducibility. So we have single chain fragment variables in our library. We produce everything in-house. We spot everything onto the microarray, and then you are going to see this is the, and our production is coming onto a table like this. So I have been working in GE Healthcare and all that kind of things where you have 23 steps of production. Here is completely different. A little bit science fiction sometimes. And what we do, we print 2,000 antibodies without cross-reaction on the surface of my pinky nail. So we then move into create our array that you saw there, a microarray with 10 to 40 antibodies on it, and then take the patient sample, the serum, so the blood, just one drop, literally a drop of blood, is placed on it, and then the sample is treated, and then it goes onto a scanner that is reading the fluorescent response, and the signal of each of this protein corresponds, so the signal of intensity of each spot corresponds to the protein concentration. You create a landscape that in the end, with 4 different algorithms, and a very advanced bioinformatics, which is also extremely important for us, it's going to give you a multidimensional space that is placing the patients on one side or the other, being healthy or sick. So I will move to the results, and I want to show you that we have run 6 large clinical studies. Over 2,500 blood samples were going through this. We worked with stages 1 to 4, with the main focus on stages 1 and 2, with, of course, match controls. And since we are Swedish, the same way as you in Norway, use a very structured and systematic approach. So we work with discovery studies, then we move on into pre-validation, and then into validation. As you can see, the bubble becomes bigger and bigger because the studies are larger and larger. To show and provide the certain probability statistical that we have to show. And the discovery studies are all published. If you are interested, just write me, I send you. They are extremely interesting. We also have now a manuscript that was submitted to Lancet just last week, and it's just summarizing this large, very large, the largest pancreatic cancer retrospective study done so far, which has about 1,400 samples with a focus on stages 1 and 2. And we found them in collaboration with Rigshospitalet and Herlev in Denmark. Thank you very much. And a fantastic biobank with extremely good clinical records, which are the most important for developing. As you see, I say training and test set. So you train your system and you test your system. And then we validated now recently, just finished last October, with samples from an American cohort. So what did we get? We get 96% accuracy, meaning that we can retrospectively detect 96% of the asymptomatic patients, stages 1 and 2, and 98% for all of them. Here you see, you are not able to visualize a 450 multidimensional space. It's not possible, only with the 4 algorithms that I don't know what the bioinformatics are doing. But this is a 3-dimensional plot where you can see the training set and the pancreatic cancer stages 1 and 2. Then on the top of it, you see the test set and how well they are separated. And in other words, we call it area under the curve, which represents the accuracy, sensitivity, and specificity. You have 1%, 1 as being the best, and you have 50, 60% as you have for PSA, for example. Here we obtain 0.96 for the stages 1 and 2, and 98 for 1 to 4. Now, the last part I have is on the clinical uses, and it's actually pretty interesting and straightforward. We look for high-risk individuals and high-risk groups. And here we find very well defined the hereditary, which is according to guidelines that are published and screening programs around the world. Then we also have found, this is called familiar pancreatic cancer, and you see there all the inclusion criteria, very much based on genetics. Then you have the diabetes type 2, which is the late onset or new onset is called after 50, that have a very much increased risk, 8 to 10 times, of developing pancreatic cancer between the first and the third year after diagnosis. After that, it's just the same as all the others. The third is vague symptoms for all the people who are coming in and having a number of criteria that are also included here. And then confirmatory diagnosis and monitoring. What are we doing here? As you can understand, we are about 25 right now in the company, so lots of work. We have a prospective longitudinal validation clinical study called PanFAM-1, where I'm going to show you in just 1 second. We work on retrospective and prospective planning of studies with NCI and with several other centers around the world for the diabetes. And then we are also looking into collaboration for a prospective validation for the vague symptoms. There are also a lot of discussions about the precancerous lesions, the ones who are very familiar with it. I can see it in your eyes. You wonder what we do there, and we do work with that as well. Now, just last, before I end, what do we do for the prospective validation? We have taken on this project where we are looking for early diagnosis through high-risk surveillance of familiar individuals. And we have been calculating with the statisticians how many people do we need to be able to get the number of events that will be necessary in such a study and the time that we need. And we have a global multi-center approach where we already have collaborations with Mount Sinai Cancer Institute in the US, University of Liverpool, Michigan, and also Madrid, Ramón y Cajal. We started the study in December, but we are still recruiting. So if you are interested, I'm very interested to get in contact with you so we can look into bringing Norwegian individuals into the study, which I think is extremely relevant and interesting. So you will remember that we have a test that is offering high accuracy in retrospective studies, 96% for early stages of pancreatic cancer. We are in a validation stage, and we are looking into starting diabetes clinical studies, vague symptoms. We work in collaboration with early detection programs in US, in Europe. We are looking to get a CE mark for the test in the same time as the global accreditation of the labs that we are now working on. And we also, you see the light there up, I see it as the regulatory approval, you know, and the possibility to get the test through reimbursement. So I'll just take you through the last and just give you a question. So I would like you to know what is asked here. If a healthy patient is coming to a doctor for a routine checkup and the doctor says, I have good news and bad news. The bad news is that based upon the latest statistics, 1 out of 2 men and 1 out of 3 women will get some type of cancer during their lives. If we wait until you have symptoms of the cancer and the cancer is advanced, you will have a 25% chance of survival. The good news is that if we detect the cancer early, you would have a 95% of surviving. And we now have a simple blood test for the early detection of the most common types of cancer. Do you want me to order the test? So, thank you.
Thank you.